Can we point to one specific general phenomenon in the body that underlies aging?
So DNA is 6 foot long. If you join your chromosomes together, you get a 6 foot per cell. So there's enough to go to the moon and back 8 times in your body. And it has to be wrapped up to exist. I actually ended up boiling aging down to an equation, which is the loss of information due to entropy. You try to copy a cassette tape, or even if you send information across the internet, some of it will get lost. That's what I think is aging. There's a biological age that you can measure. Unfortunately, the test is many hundreds of dollars right now. So I want to democratize this test so that everybody has access to a score for their health that can predict not just their future health and time of death. And I'm building a system that will point people in the right direction, that will improve not just their health now, but 10, 20, 30 years into the future. And we can measure that, because if you don't measure something, you can't optimize it.
Can we point to one specific general phenomenon in the body that underlies aging?
Yeah, well, that's contentious because scientists like to come up with new hypotheses. It's how they build their careers. But fortunately, during the 2000s, we settled on 8 or 9 major causes of aging. We called them hallmarks because causes was a little bit too strong. But these 8 or 9 causes, at least for the first time, allowed us to come around and talk together. We put them on a pizza, so everyone got an equal weighting, equal slices. But before that, by the way, we were trying to kill each other in the field. It was horrible.
Interesting that you guys work on aging and trying to kill each other.
Yeah, isn't it? Well, kill each other's careers. I mean, I like to think I was fairly generous, but I was one of the kids, and the old guard really didn't like the new guard. We just came along in the 1990s and 90s and said, free radicals don't do much. There are actually genes called longevity genes, and that caused a whole ruckus. And there was this competition for what never happened, which was a Nobel Prize for this. And it just led to a lot of competition. I would go to meetings and people would shout at each other and just backstab. It was horrible. But then fortunately in the 2000s, we rallied around this new map of aging with these causes or hallmarks. But I think that there's one slice of the pizza that is way larger than the others. And we can get to that, but that's the information in the cell that we call the epigenome.
Well, tell us a little bit more about the epigenome. Frame it for us, if you will, and then we'll get into ways that one can adjust the epigenome in positive ways.
Yeah, so in science, what I like to do, I'm a reductionist, is to boil it down. And I actually ended up boiling aging down to an equation, which is the loss of information due to entropy. It's a hard thing to overcome the second law of thermodynamics. That's fair. But this equation really represents the fact that I think aging is a loss of information in the same way that when you Xerox something 1,000 times, you'll lose that information, or you try to copy a cassette tape, or even if you send information across the internet, some of it will get lost. That's what I think is aging. And there are 2 types of information in the body. There is the genetic information, which is digital, A, T, C, G, the chemical letters of DNA. But there's this other part of the information in the body that's just as important. It's essential, in fact, and that's the systems that control which genes are switched on and off, in what cell, at what time, in response to what we eat, etc. And it turns out that 80% of our future longevity and health is controlled by this second part, the epigenetic information, the control systems. I liken the DNA to the music that's on a DVD or a compact disc. For the younger people, we used to use these things.
I recall.
Yeah. And then the epigenome is the reader that says, okay, in this cell, we need to play that set of songs. And in this other cell, we have to play a different set of songs. But over time, aging is the equivalent of scratching the CD and the DVD so that you're not playing the right songs. And cells, when they don't hear the right songs, they get messed up and they don't function well. And that is what I'm saying is the main driver of aging. And these other hallmarks are largely manifestations of that process.
Can we go a little deeper into what these scratches are? Is it the way that the DNA are packed into a cell? Is it the way that they're spaced? What are the scratches that you're referring to?
So DNA is 6 foot long. So if you join your chromosomes together, you get a 6 foot per cell. So there's enough to go to the moon and back 8 times in your body. And it has to be wrapped up to exist inside us. But it's not just wrapped up willy-nilly. It's not just a bundle of string. It's wrapped up very carefully in ways that dictates which genes are switched on and off. And when we're developing in the embryo, the cell marks the DNA with chemicals that says, okay, this gene is for a nerve cell. You cell will stay a nerve cell for the next 100 years, if you're lucky. Don't turn into a skin cell. That would be bad. And those chemicals, there are many different types of chemicals, but one's called methylation. Those little methyls will mark which songs get played for the rest of your life. And there are other marks that change daily. But in toto, what we're saying is that the body controls the genome through the ability to mark the DNA and then compact some parts of it, silence those genes, don't read those genes, and open others, keep others open that should stay open. And that pattern of genes that are silent and open, silent, open, is what dictates the cell's type, the cell's function. And then the scratches are the disruption of that. So genes that were once silent, and you could say it's a gene that is involved in skin, it's starting to come on in the brain, shouldn't be there, but we see this happen. And vice versa, the gene might get shut off over time during aging. Cells over time lose these structures, lose their identity, they forget what they're supposed to do, and we get diseases. We call that aging. And we can measure that. In fact, we can measure it in such a way that we can predict when somebody's going to die based on the changes in those chemicals.
Along the lines of supplementation, what about NMN, NR, and B3 niacin? I want to know what you do. I also want to know what I should do. And I think most people want to know what they should do. I mean, these are molecules that impact the sirtuin pathway, impact the pathways that control aging or rates of aging in the epigenome. How do they do that? And how does one incorporate that into a supplementation protocol, should they choose to do that?
Well, disclaimer is that I don't recommend anything, but I talk about what I do. So a bit of scientific background, these sirtuin genes that we discovered first in yeast cells when I was at MIT, and then in animals as I moved to Harvard in the 2000s. And one of my first postdocs, actually literally my first postdoc, Chaim Cohen, published a great paper just a couple of months ago and found that turning on the sirtuin 6 gene, remember the 7, number 6 gene is very potent. It extended the lifespan dramatically of mice that he engineered, both males and females, which is great. So what you want to do is naturally boost the activity of these sirtuins. They are genes, but they also make proteins. That's what genes typically make, or encode. And then those proteins take care of the body in many different ways, as we've discussed. So how do you turn on these genes and make the proteins they make even more active? You want to rev up that system. So exercise will do it. Fasting will do it. What about supplementation? Well, the first activator of the sirtuins that we discovered that acts on the enzyme to make it do a better job of cleaning up the body and protecting was resveratrol. We looked at thousands of different molecules, eventually tens of thousands. And the one that was the best was resveratrol in the dish. And then we gave it to little organisms, worms, and then flies and mice, eventually humans. And we saw that it activated that enzyme. So resveratrol is one way to activate it. You can think of it as the accelerator pedal on a car. It revs it up. But there's something else that the sirtuins need to work, and that's NAD. NAD is a really small molecule, little chemical in the body that we need for life. It's used by the body for chemical reactions, 400 different reactions in the body, and without it, you're dead within seconds. You need NAD. The problem that we've seen is that NAD levels decline as you become obese, as you get older, if you don't ever get hungry. And the body not only doesn't make enough of it, it's chewing it up as well. There's an enzyme called CD38 that Eric Verdin over at UCSF showed chews up, he's now at the Buck Institute in California, chews up NAD as you get older. So it's a double whammy. You don't make as much, you chew it up, which is really bad because what we've shown in my lab, and so have others, is that NAD levels are really important for keeping those sirtuin defenses at a youthful level. And you can give a lot of resveratrol, but if you don't have the fuel, you're basically accelerating a car that doesn't have enough gas. So you want to do both, and that's what I do. I take a precursor to NAD called NMN, and the body uses that to make the NAD molecule in one step. And so I know from measuring dozens of human beings that if you take NMN for the time period that I do, I've been taking it for years, but if you take it for about 2 weeks, you'll double on average, double your NAD levels in the blood. Okay, that's not public information. That's from clinical trials that are not yet published over the last 2 years. There are other ways to increase NAD levels in someone like me who's getting older, I'm 52 now. You can take NR, which is used to make NMN, which is used to make NAD. And both NMN and NR are sold by companies in the US. NR lacks the phosphate. Phosphate's a small chemical that the body needs. You've probably heard of the atom phosphorus. Let's go back one step. How do you make NR? NR gets made from vitamin B3 often. You can also find it in milk and other foods. But sometimes people ask me, why don't you just take vitamin B3 and won't that just force the body to make NAD? And the answer is no, it doesn't work very well. We know this just by doing the experiment. But the reason I think is that NAD is I said it's a small molecule, but relative to vitamin B3, it's big. It's got those phosphates on there. It's got a sugar. It's got the vitamin B attached. So you've got all these components that come together to make this very complicated little molecule called NAD. And when you give NMN, it contains all three components that the body needs to make NAD. If you give NR or just vitamin B3, which is an even smaller molecule, the body has to find these other components from somewhere else. So where do you get phosphate? Well, the body needs it for DNA, needs it for bones. So high doses of something that requires additional phosphate makes me a little concerned. And we have compared NMN and NR head-to-head in mouse studies. For instance, NMN, we've shown in a Cell paper a few years ago, makes mice run further. Old mice can run 50% further because they have better blood flow, better energy. NR at the same dose did not do that. In fact, it had no effect.
I see. Dosage-wise, if I were elect to take NMN in supplement form to increase my NAD levels and presumably slow my aging, how much NMN should I take? What's the protocol that you do? And are the various forms that are out there, are some better or some worse?
Well, I'm always happy to tell you what I do and what my father does, my 82-year-old father. We take a gram of NMN every day.
So it's a gram of resveratrol and a gram of NMN.
Right.
Okay, 1,000 milligrams.
Now, another important point, which is I'm not the same as everybody else. I have a different microbiome, age, sex, right? And so I've been measuring myself. And so I know if something's, or I think I know if something's making me better or worse based on measuring 45 different things. So I just want people to be aware that what I do may not perfectly work at all for others. But I have studied, as I said, dozens of people who take NMN at a gram, sometimes 2 grams. And I know by looking at all those people that without any exceptions, that if you do what I do, your NAD levels go up by about 2-fold or more. And so I do that every day, the 1,000 milligrams. Now people sell it. Now I never get into brands and all that. First of all, I don't have the time to measure products. I don't know. Though I do want to say I'm working on a solution for people to know what works and what's real and what isn't. But I'm not there yet. And in the meantime, I would say, if you do want to buy this, let's say you want to buy NMN, look for a company that is well established, that has high levels of quality control. Look for 3 letters, GMP, which is good manufacturing practices. And so that means they make it under a certain level of quality control. You're not going to find iron filings in there, and it probably has the stuff in it that they say it does. So that's all I can say right now. I'm working on something that's going to be much more helpful. But overall, make sure it's white, crystalline NMN, and that to me it tastes like burnt popcorn.
You crack open the capsules and you'll take a little sample to make sure it tastes like burnt popcorn.
Well, when I'm making my capsules, I'll taste it, and I do a lot of quality control on the stuff that I take.
Do you take that gram all at once with the resveratrol, or do you take it spread throughout the day?
It's all in the morning for those things. So if I take metformin, it's NMN and the resveratrol all together, and there's a good reason for that. It's all scientific. I try to be. The levels of NAD go up in the morning in our bodies naturally. Our bodies actually have a cycle of NAD. It's not steady.
It's circadian?
It's circadian. In fact, NAD controls your clock. This was shown by Shin M.I. and colleagues in a nice science paper about a decade ago, that if you disrupt the NAD cycle, which is controlled by the sirtuin gene that we worked on, that is what's telling your body, oh, it's time to eat, it's time to go to sleep. And if you take the NMN late at night, for example, you can disrupt your circadian rhythms.
Interesting.
Conversely, when I travel and I want to reset my clock to the time zone, I will take a boost of NMN in the morning and I feel great.
Does this protocol for you, does it produce any immediate effects of increased energy, et cetera? You mentioned that one would, if it's right for them, would have to take it for at least 2 weeks to start to see the NAD levels increase. At that point, when NAD levels increase, could one possibly expect an increase in overall energy, focus, et cetera?
Well,
I realize we're not making promises here, but I'm just wondering whether or not the only measure of whether or not this protocol is working is whether or not you die at age blank or blank plus 20. And of course, once you're dead, you can't really know if you would have lived longer if you'd done something differently, and vice versa.
Sure. Well, there was a study, again, by Shin M.I., my good friend at Washington University in St. Louis, that showed that it improves, remember this, insulin sensitivity, which is a good thing. But you can't know your insulin sensitivity unless you're measuring have a glucose monitor on your arm.
Do you have one on right now?
No. No, I used to. I learned a lot.
Yeah, last time I saw you, you had this thing. It looks like a small leech, not a large leech, and it was measuring your blood glucose.
They're very informative because you learn what your body reacts to, and grapes were really bad. Rhonda Patrick agrees with that. But the issue was what? Where were we, Andrew?
The issue is whether or not you can expect any immediate effects on energy, vitality, focus, just even subjective.
So what do you feel is the question. And anecdotally, because I've been taking this for a long time, if I don't take it, I start to feel 50 years old. It's horrible. I can't think straight. It may be placebo, but who knows? But what we're doing now are very careful clinical trials. We've done the safety for 2 years, and we're now treating elderly patients at Harvard Medical School with some wonderful colleagues. And those people are actually going to be, and currently in MRIs, so you can measure the energetics and the NAD levels in their legs as they exercise in real time. And that will tell us if what we see in the mice, this increased endurance, actually works. In the meantime, it's fun to talk about anecdotes. I have a number of athlete friends, some of which have increased lowered their time in marathons, for example. There's a good friend of ours in our circle that is winning marathons at age 50 now, and he attributes that to the protocol that he's on.
I haven't started taking NMN, but I'm planning to do that when my next birthday arrives, which is in a couple months. But I do experiments on my sister and have for years. I have a sister who's 3 years older than I am, who is very enthusiastic about these protocols. And I'll tell you that after reading your book, I started purchasing for her and giving her an NMN supplement. And she claims, and I believe her, she has a quite sensitive system and she's very tuned into it. She feels far and away better when she takes it as opposed to when she doesn't. And I've done the control experiment of removing her supply and then giving it back to her and this kind of thing. Zooming way out, what are the behavioral tools that one can start to think about in terms of ways to modulate these, basically the way that DNA is being expressed and functioning? I've heard you talk before about hormesis of other sorts, cold exposure. We talked about fasting. We talked about exercise in broad terms, but what about any evidence, if it exists, as to whether or not aerobic training versus weight training, these sorts of things? In other words, what are the sorts of things that people can do to improve the sirtuin pathway? And I realize that there are caveats. We can't go directly from a behavior to sirtuins, but in the general theme, what can people do? What do you do?
Right. Well, we know that aerobic exercise in mice and rats raises their NAD levels, and their levels of SIRT, one of the genes goes up, two actually, number 1 and number 3. What we don't know yet is what type of exercise is optimal to get them to change. We will learn. We're doing work. Now it's revealed that we're doing work with the military in the US to try and understand that kind of thing. And I'll always tell you and the public when I don't know something. I'm not going to extrapolate. But what do? I base my exercise on the scientific literature, which has shown that maintaining muscle mass is very important for a number of reasons. The two main ones are you want to maintain your hormone levels. I'm an older male, losing my testosterone and muscle mass over time. And by exercising, I will maintain that and have. In fact, I probably haven't had a body like this since I was 20. So that's one of the benefits of having this lifestyle.
Sorry to interrupt you. You know, we did an episode on hormones, and there are data in humans that show that there are some males in their 80s and 90s where their testosterone is equivalent to the average of 25 and 30-year-olds. I can get you that information. It is really impressive studies. Unfortunately, they didn't include a lot of information about the lifestyle factors, et cetera. But this idea that testosterone goes down with age, it might be the trend, but it's not necessarily a prerequisite.
Right. I believe in naturally increasing and maintaining these hormone levels. And I've been measuring them for a long time. And I could see, for me, my testosterone levels were going down.
And then you got tenure and they went back up again.
No. I actually became complacent and it was the worst. Actually, my age changed in the wrong direction after that because I was relaxed.
Interesting.
And not worried about the future. But then I got serious and I actually, according to the Inside Tracker algorithm, got my age down from 58 to 31 in a matter of months. So that was a big drop. And I've been getting steadily younger over the last 10 years, according to that measurement, the blood test.
What about estrogen? Because women are different in the sense that the number of eggs and the ovaries change over time, right? Do you think that they can maintain estrogen levels over longer periods of time using some of these same protocols?
Well, yeah, I get into trouble from a certain university when I talk about this too much.
About estrogen?
Just about fertility and long story. That if I don't want to get too much into the anecdotes, but I'll tell you the science, which is that if you take a mouse and put it on fasting or caloric restriction for up until the point where it should be infertile. So that's about it. At a year of age, a mouse gets infertile, female mouse.
Due to fasting? Or no, due to simply to aging?
Due to aging.
Okay.
Due to aging. The fasting, it's not an extreme fast, it's just less calories.
Got it.
Then you put them back on a regular food and they become fertile again for many, many months afterwards. So the effect on slowing down aging is also on the reproductive system.
Interesting.
And so that I wouldn't say to any woman, I wouldn't think that they should become super skinny to try and preserve fertility. That's not what I'm saying. But these pathways that we work on, these sirtuins, are known to delay infertility in female animals. Case in point, I'm one of the lead authors on a paper where we used NMN. Remember, this is the gas, the fuel, the petrol for the sirtuins. We gave old mice, one group of mice was 16 months old. Remember, they became infertile at 12. Gave them NMN, and I think it was only 6 weeks later, they had offspring. They became fertile again, which goes against biology, textbook biology, which is that female mammals run out of eggs. Turns out that's not true. You can rejuvenate the female reproductive system and even get them to come out of mousopause, as we call it. So that's a whole new paradigm in biology as well.
That's super interesting. Sorry to interrupt you, but I'm reminded by a set of studies that were done by your former colleagues, because they're no longer there. David Hubel and Torrance and Wiesel, my scientific great-grandparents, won the Nobel Prize for discovering what are called critical periods, this phase of early development when the brain is extremely plastic. And a big part of their work was to show that after a certain point, the critical period shuts down. Essentially, the brain can't change, or not nearly as much. And then people came along later and showed that you could open up these critical periods again, but very briefly, and it takes a very specific stimulus, essentially, high degrees of focus, etc. However, there's a well-known phenomenon in this literature where if you take an animal, and to some degree, this is being shown in humans as well, and you let them pass through the critical period, but then you essentially sensory deprive them. You take away experience, you close both eyes. You essentially reopen the critical period. So it seems like, and I couldn't help but mention this, that there's this parallel between what we're talking about here with fertility and neuroplasticity, where yes, there's a timer where certain things are available to the organism early in life, and then they tend to taper off. It's not an open and shut, but they taper off. But then a deprivation can actually reactivate the availability of that process. Forgive me, I just couldn't help but mention it. But to me, both of those things are associated with youth, fertility and neuroplasticity. And so I think that it'd be so interesting. I'd love to collaborate with you on this to explore how neuroplasticity might actually be regulated by these things like the sirtuins. You reminded me of something that I meant to ask earlier, that obesity reduces NAD levels and accelerates aging. How? I mean, okay, so again, this is the scientist in us. So someone's carrying a lot of excess adipose tissue, subcutaneous and visceral fat. But why should that reduce NAD in any ways that are independent of effects on glucose and insulin? Is there something direct about white adipose tissue? And the reason I ask this is not simply to dig into mechanism alone, but I think there are really interesting data now that fat actually gets neural innervation. I mean, it's not just stored fuel. It's stored fuel that's acting as an endocrine organ, essentially. So, why would being fat make people age faster?
Yeah, that's a question that is so obvious, but so few people ask it. That's what makes you a good scientist. And so that we don't know, but I'll give you my best answer, which is that obesity comes along with a lot of problems that include a lot of senescent cells in fat. If you stain old fat for senescent cells, it lights up. Mm-hmm. And when you kill off those cells, at least in mice and maybe in humans, it looks like the fat is less toxic to the body because those senescent cells in their fat are secreting these inflammatory molecules that will accelerate aging, as we now know. You talk about the sirtuins and NAD. So if we just look philosophically at why this would be, the sirtuins only like to come on or get activated when the body is under adversity. And if a cell is surrounded by fat or contains a lot of fat, it's going to think times are good, doesn't need to switch on. So that's the evolutionary argument. Mechanistically, we don't know, but it could have something to do with the response to glucose, which then responds to the sirtuin gene. But that hasn't been worked out very well.
And is there any evidence that leptin, this hormone from fat, can actually interact with the sirtuin pathway?
I don't recall seeing that.
Maybe I could do a sabbatical in your lab, and that'd be a fun one.
Definitely.
Because leptin during development is what triggers the permission for the hypothalamus to enter puberty. Right.
Yeah.
This is why kids that eat a lot when they're young and get overweight will also start to go and undergo puberty more quickly, although they have reproductive issues later.
Well, yeah, we should study the hypothalamus together because the hypothalamus can control the aging of the body.
The most interesting part of the brain.
For sure.
Yeah,
for sure.
Absolutely.
If you turn on the SIRT1 gene, the sirtuin gene that we work on in the hypothalamus, it actually will extend lifespan. Also, it's been shown by Dongsheng Kai at Albert Einstein College of Medicine that if you inhibit inflammation in the hypothalamus in a mouse, it will increase or maintain the expression of what's called GnRH, which is the hormone that he found actually controls longevity in the mouse in part. And so keeping inflammation down in the hypothalamus is sufficient to extend the lifespan of animals. And I reviewed that paper for Nature about 7 years ago. And that was the first demonstration that the hypothalamus is one of the leading regulators of the body's age.
I find this fascinating. GnRH, for those of you that don't know, actually comes from neurons in the hypothalamus that then literally reach down into the pituitary and trigger the release of all the things that control fertility, luteinizing hormone, follicle stimulating hormone, et cetera. It's such a powerful set of neurons, and it's never really been clear what at a behavioral level triggers the release of GnRH. There's all the stories about pheromones and timers in puberty, et cetera, but environmental conditions and dietary conditions and behaviors that can control GnRH release, I think, is an incredible area for exploration. I'd love to do that sabbatical, by the way. Recently, I've noticed that you've opened up a sort of an email/website that people can access, to get some information about their own health and rates of aging. Tell us about that and what's being measured and what is this test that you've been working on secretly and now soon not so secretly?
Yeah, well, what I want is a credit score for the body to make it easy for people to follow their health. And there is a number. There's a biological age that you can measure. Unfortunately, the test is many hundreds of dollars right now. But in my lab, we've been able to bring that down a lot. And so I want to democratize this test so that everybody has access to a score for their health that can predict not just their future health and time of death, but to change it. And I'm building a system that will point people in the right direction and give them discounts for certain things that will improve not just their health now, but 10, 20, 30 years into the future. And we can measure that and very cheaply keep measuring it to know that you're on the right track. Because if you don't measure something, you can't optimize it. And so this is the biological age test. We've developed it. It's a simple mouth swab. We're rolling it out. We're building the system right now. And there is a sign-up sheet because a lot of people want to get in line. Go to drsinclair.com. You can get on that and you'll be one of the first people in the world to get this test and see what we're doing.